Okay. Hello everyone. Myself Dr. Kushal Salunke, Junior Resident and Demonstrator in Department of Biochemistry, GMARS Medical College, Gotri, Vadodara. Okay. So in this video, we will be having a discussion of qualitative analysis of carbohydrates. Okay. This is your very first practical that you are going to perform in your UG biochemistry lab. Okay. After the introduction has been done. Right. So this is very basic practical and yet important for your Viva examination also. There are so many questions that can be asked in your Viva examination. We can put any test in our spotting also. So this is very important topic. Okay, as an exam point of view. Okay, you might not be asked performing this practical in your practical examination, but shortly the examiner will ask you the Viva questions from this topic. Okay, what I have done, I have covered mostly all the questions that can be asked in your Viva examination also. Okay, and I have covered and I have simplified the theory behind it also. Okay, so let's start discussion of qualitative analysis of carbohydrates. Okay, so what you know from your theory classes, what are the carbohydrates? So carbohydrates are the organic compounds that will compose of carbon, hydrogen and oxygen, and generally which is in a ratio of Cn and H2O(n). Okay, carbon and the water molecule are same in the number. Okay, and hydrogen molecule will be double as the number of carbon molecule, so they are a major source of energy. Okay, in all living organism, you know, they are classified according to monosaccharides, that is single sugar unit, that is glucose and fructose. Disaccharides, there are two monosaccharide units which is joined by glycosidic bond. Example is sucrose and maltose. I hope this thing is very clear from your theory classes. So this is also a revision of your theory class also. Polysaccharide, they are long chains of monosaccharide. Example will be starch, glycogen and cellulose. So while we are performing the qualitative test on these particular substances, they will detect the presence of carbohydrate in any sample. You can take a urine sample. In our lab, they will be coming a urine sample and we have to perform the Benedict's test on it. Okay, to detect a carbohydrate or sugar. Okay, whenever a pediatrician is suspecting any inborn error of metabolism or essential pentosuria and something, so they will send a sample to our biochemistry lab and we will perform the test, that is usually basic test we are going to perform, that is qualitative analysis of this particular carbohydrates. Okay, moreover this test helps in differentiating the types of monosaccharide, disaccharide as well as polysaccharide. They also distinguish between the reducing and non-reducing sugar, and this test also identify the aldose sugars and the ketose sugar. Aldose sugar mein we have aldehyde group and ketose sugar mein we have ketone group. Okay, you know what is the functional group? So functional group mein we have carbonyl group as well as hydroxyl group. That's why carbohydrates are called polyhydroxy compound. Okay, they are polyhydroxy compound. So one or more group will be there. Okay, in the carbohydrate structure. So the qualitative analysis of carbohydrates utilize a specific color reaction. They will utilize a specific color reaction. Okay, based on that first, that is dehydration reaction. So whenever you treat the carbohydrate in the acidic condition, so acid are dehydrating agents, so they will yield a furfural derivatives and they will yield a specific color. What color we will be seeing in a subsequent part. Okay, second reaction, oxidation reduction reactions are there. Okay, so these oxidation and reduction reactions, one example is Benedict's test, and they will give a colored precipitate. Okay, complex formation, starch and iodine will form a complex and that will give a jet blue color. Okay, on iodine test, and some shows isomerization also, that is aldose and ketose interconversion under the alkaline condition. Okay, so we have oxidation reaction, dehydration reaction, complex formation, and we have isomerization reaction also in this particular part. Okay, right? So now we will discuss, there are total 7 tests that we are going to discuss in detail. Okay, which principles and biochemical aspects and some applied aspects also we will touch. Okay, so first test I want to discuss with you is a mollusk test. Okay, this is a general test for all the carbohydrate molecules. You should know any carbohydrate molecule will show this test positive, right? So what is the principle? So under concentrated sulfuric acid or under the concentrated acidic part, very acidic pH, carbohydrates will go acid catalyzation dehydration reaction, acid catalyzed dehydration reaction. Okay, so whenever you are treating or you are taking a pentose sugar which undergoes acid catalyzed dehydration, it will yield a furfural derivative, and hexose sugar will yield 5-hydroxymethyl furfural. Both will give a one color. Okay, so furfural derivatives then condenses whenever you condense with alpha naphthol, a phenolic aromatic compound, via electrophilic aromatic substitution. A purple colored complex is formed at the interface. So whenever you are treating the alpha naphthol with acidic condition, this will form this particular thing. Okay, so when you treat glucose with sulfuric acid, they will form, it is a hexose, so it will form 5-hydroxymethyl furfural. When you add alpha naphthol to it, it will call a violet color complex. Okay, so what is a biochemical interpretation in it? So carbohydrate or carbohydrate moiety, that is glycoprotein and glycolipids, also will show this test positive. So this is non-specific test for carbohydrate, because both reducing as well as non-reducing sugar will yield this test positive. Okay, apart from it, some conjugate molecules like proteins which have the carbohydrate moiety, that is glycoproteins, mucopolysaccharides, also shows this test positive. Mucopolysaccharides are present in the mucus of the gastric juice. So gastric juice, when you take a fresh gastric juice, okay, from rice tube, it will also show this moles test positive. Okay, so this is non-specific test. What extra information you should take care, that is the reaction depends on furanose ring dehydration capacity. How much dehydration it will perform, that will decide. Okay, compound like glycoprotein and nucleic acid, due to nucleic acid, why, due to ribose and deoxyribose, also show this test positive. But when you overheat this particular test, when excess of dehydration is performed, when you add excess of acid, they will leads to charring, and charring will yield a brown color. Okay, sugar will be charred. So clinically you can screen this test for carbohydrate presence in a biological fluid before you add any quantitative analysis to it. Okay, right, so you can see this very nicely formed ring. Okay, very nicely formed ring in a purple color. Okay, it is very nicely formed ring, right, in a purple color. You can see, I am zooming it, you can see this very nicely formed ring at the junction of two solutions. Okay, right, you can see slightly greenish tinge above and below the ring. Why this is there, we will be discussing in a subsequent part. When you have performed the practical also, you know there is a greenish tinge sometime happens around the ring. Why, we will discussing it. You can see, and you can compare the different solutions. Fructose will also show, glucose, sucrose and starch is also showing, so all will give the purple test positive. Okay, and you can see the control will show a negative test. Okay, right, now see this, a green ring in Molly's test indicates a partial degradation or mixed reaction during dehydration of carbohydrate under concentrated acid. It usually arises due to overheating or partial charring of sugar. Okay, when you add sulfuric acid in more quantity, it will char the sugar. Okay, excessive dehydration. Also, mixed furfural derivatives from pentose and hexose also yield a greenish color, greenish tinge to it. Okay, so whenever there's two or more type of sugar in it, that also containing the in a solution which is also yielding the green color. Impurities also leading to chromophore blending. Okay, that is due to impurity. Okay, maybe you sometime any examiner you can they can ask this particular question when you have your viva at excellent level. Okay, so whenever you are performing a Mollis test on biological sample, always layer the acid very slowly, not direct mixing. Use freshly prepared alpha naphthol to avoid green contamination ring. And a distinct violet ring confirms a carbohydrate. A greenish ring still suggests a carbohydrate present, but you have done an impure reaction condition. Okay. Now let's see the some Viva questions. Okay, so you may ask why molystase is a general carbohydrate test, because all carbohydrates can form furfural derivative under dehydration which reacts with alpha naphthol. Okay, why glyceraldehyde does not give this test positive? Being a small sugar, glyceraldehyde is a 3-carbon sugar, and 3-carbon sugar it going to decompose when you are treating with a strong acid, so it won't yield a positive test because sugar is decomposed na, that's why it won't yield. Okay, right, I hope this thing is very clear. Okay, okay, so you understood the concept. right? Now, why should the acid is added slowly? This is also a very important Viva question. Okay. So to prevent violent charring and mixing, the ring form only at the interface. You should add the acid very slowly. Okay. Can proteins give this Molecular stress positive? Yes, glycoprotein is the protein which gives the Molecular stress positive due to carbohydrate content. So why nitric acid is not used? Okay. Sometimes I have observed students are doing practical and on the concentrated solution rack there is a concentrated nitric acid solution just beside the H2SO4, and many times the labels are not visible, so they put HNO3 instead of H2SO4, and therefore they came to me and they are showing fully dark brown colored complex, and they are saying sir nahi aa raha hai Mollis test. Okay, why this is happening? So nitric acid, you should note, is a strong oxidizing agent. Okay, and they will destroy the sugar only, so it won't give any test. Okay, apart from it, whenever you are adding the alpha naphthol to it, it also destroys the alpha naphthol also. Okay, so this is the thing, that's why you are not using, and you should use carefully. Okay, because both are concentrated acids. Okay, right, you have to ask lab technician what is H2SO4, then you are using. Okay, then you have to use it. Now this is also second test. Okay, this is iodine test. Okay, this is an iodine test, right? So what is the principle of iodine test? क्या होता है प्रिंसिपल आयोडीन टेस्ट का? So whenever iodine reacts with the helical configuration of amylose, it is alpha 1,4 linkage. Amylose contains alpha 1,4 linkage. Okay, so whenever alpha 1,4 linkage is there, right? Okay, so whenever alpha 1,4 linkage is there, they will form a polyiodide chain with helical cavity. The color intensity is due to charge transfer between starch helic and polyiodide compounds or polyiodide ions, right? So what polysaccharide? Suppose we take amylose, which is a linear. Structural feature is a tight helix, so it will yield a blue color. Amylopectin, if you are taking this branch and shorter helix, so it will yield a violet color. And glycogen, important point to note, which is highly branched and we have short helix, it will yield a reddish brown color. I'll show the colors first. Okay, then we'll discuss subsequently what are the dynamics of reaction. You see this. Okay, this is, you can see the dark brown or red color. Okay, you can see this is a blue, and you can see this is a violet color. Okay, so blue color will be formed by starch, violet dextrin, and reddish brown will be glycogen. Okay, so the color disappears on heating due to helical dissociation and reappears on cooling as helix reforms. There is no reaction with cellulose because cellulose contain beta 1,4 linkages and monosaccharides does not have any helix, so these two will not yield any test. Okay, when you are performing the test on the glucose, it won't give a blue color, right? So clinical insight, it will use to assess starch digestion or hydrolysis completion. So color disappears as hydrolysis proceeds. Okay, it will indicate a polymeric nature of carbohydrates. So what are the Viva questions? So why does glycogen need a reddish brown color? Glycogen has short helix and hold fewer iodine molecules. Okay, and alternating electronic absorption is there. So why reversible with heat? Because why it is reversible with it? So non-covalent van der Waals and hydrogen bond stabilizes the complex, and this heat disrupts this complex temporarily disrupt karta hai, not permanently. Okay, van der Waals forces and hydrogen forces exist between these non-covalent bonds. Okay, right, you can see on the bread, yeah, there is a starch. Okay, and when you put iodine on the bread, it will yield such dark brown color, dark blue color. You can see some kind of YouTubers and Instagram viewers that paneer. Okay, पनीर पर भी ये टेस्ट करते हैं, and it will yield a blue color, and some will show it is fake paneer and all. Okay, this is totally nonsense. Okay, right, anything which contains starch will show this test positive. Okay, right. Now Benedict's test में for reducing sugar, it is that. So theory behind the Benedict's test is, Benedict test identifies the sugar, okay, those with free aldehyde group and free ketone group. Okay, this is important point. Free aldehyde and free ketone group होना चाहिए, otherwise it won't give this test positive. Okay, many a times in a Viva question they ask a reagent composition. Okay, first of all, I want to discuss that Benedict's reaction reagent will contain a cupric ions. Okay, so copper sulfate is the source of cupric ions. Sodium carbonate creates an alkaline medium, and sodium citrate keeps copper in a solution by complexation. Okay, how will you perform this procedure? You have to add 5 ml Benedict's reagent to a test tube first of all. Then you have to add 0.5 ml of test solution. You have to boil it and up to 2 3 minutes, and then you have to cool it down. So color interpretation, green is very small amount of sugar, moderate में yellow, orange में high, brick red is very high. So green में it is single positive, yellow में it is double positive, orange में it is triple positive, and brick red में it is strongly positive, that is four positive. Okay, I'll show you the photograph. See this. This is absolutely negative test. This is single positive, this is double positive, and this is, sorry, this is triple positive, and this is four positive. Okay, I hope this thing is very clear. So why Benedict's test is semi-quantitative test? Okay, it is semi-quantitative test because color intensity and precipitate amount will be roughly correlate with sugar concentration. Okay, all monosaccharides and disaccharides except sucrose will respond positively, and keto sugar like fructose react after tautomerization. This is important point. Okay. So why does sucrose does not give Benedict test? Because it lacks free carbonyl group. Both anomeric carbons are involved in the glycosidic linkages. Okay. That's why sucrose won't give the test. How does fructose act as a reducing sugar? So under alkaline condition, it gives a tautomerized product to glucose and mannose aldoses, which reduces copper plus 2 ions. Right? I hope this thing is clear. Under alkaline medium, you can see copper plus 2 ion will go, and here if you find the oxidation number, it is positive, single positive. So copper plus 2 ion का reduction होता है, right? This thing is very clear, right? Okay. Okay, now coming to next question, why sodium citrate is added in a reagent. Okay, so it complexes the copper plus 2 ions, okay, copper plus 2 ions to prevent copper hydroxide precipitation before the actual reduction procedure. Okay, in this diagram, in this image, you can see when we have added water and Benedict's reagent, it will show a negative test. In the test tube number 3, we have added sucrose and Benedict's reagent. It is also showing a test negative, but you can see glucose and Benedict's will show this particular test positive. Okay. Now let's discuss fourth test, that is Barfoed's test. Okay, Barfoed's test. It is for monosaccharide. Okay, theory behind it is similar to Benedict's, but it is done in acidic medium. You have to note this particular that it is done in acidic medium. Okay, I hope this thing is very clear. Okay, only strong reducing sugar, that is monosaccharides, can reduce copper plus to copper plus rapidly in this condition. Okay, so you know this aldehyde group containing in a carbohydrate that is on reducing sugar, when it will react with copper plus 2 in a acidic medium. So we are using here we are using acidic medium. So it will form this particular salt or this particular acid. Okay. So we have to add 2 ml of Barfoed's reagent plus 2 ml of test solution. You have to boil it for 30 to 60 seconds, and you have to cool it under the water. So you can see very nicely from precipitate of copper oxide. Okay, within 1 to 2 minutes. So that will show a monosaccharide is present. Like what monosaccharides, glucose and fructose can be present, right? Glucose and fructose might be present on monosaccharide. So inference from this you can take a monosaccharide is present. Concept note you should know. Disaccharides reacts very slowly. Why? So you have to require a prolonged boiling, okay, to break down the bonds between the disaccharides. So there is a time for appearance of precipitate that will differentiate the monosaccharide and disaccharides. Okay, this is very important bio question. You can ask this particular, you may ask this question. Okay, and this is also a very good question. Now let's see why Barfoed is performed in acidic medium. Okay, to slow down the reaction. Okay, to slow down the reduction so that only monosaccharide can react quickly. Okay, that we can show. Can you differentiate monosaccharide with disaccharide by using Barfoot's test? This is also a very good question. So by reaction time, monosaccharide will give rapid precipitate within 2 minutes. Disaccharide, you have to boil it for more than 5 to 10 minutes. Okay, then they will show the reaction, right? So what happens on prolonged heating? Disaccharide hydrolyzes to monosaccharide and it will lead to false positive test. Okay, see this. You can see very clearly formed precipitate at the bottom, right? You can see very clearly formed precipitate at the bottom. See this red precipitate. Okay, okay, I hope this thing is very clear, right? Now let's discuss ahead. You can see Barford stress on monosaccharides, brick red precipitate formed by the glucose. Okay, you have to require a prolonged heating for lactose and maltose. Okay, and scanty brick red precipitate will be formed by sucrose and no बिबिटिना कंट्रोल, राइट? एकदम स्कैंटी रेड फॉर्म होता है सुक्रोज के अंदर. ओके, if the concentration is beyond 1%, then only, otherwise it won't give test. Now let's discuss Saliminoff test for ketose sugars. So theory is under acidic medium, ketose will dehydrate faster than aldoses, forming hydroxymethyl furfural derivative, which will react with resorcinol to give cherry red complex. So whenever I am taking a fructose, that is a ketose sugar, when I react with acidic condition, it will form hydroxymethyl furfuran and resorcinol, and which ultimately hydroxymethyl furfural plus resorcinol will give a cherry red product. Okay, and this is the color. So how will you perform the test? You have to mix 1 ml of test solution with 1 ml of Saliminoff's reagent, and Saliminoff's reagent, the content would be, you may ask in your viva, resorcinol and concentrated HCl. So you have to boil it for 30 seconds and let it be cooling down. Observation will be cherry red precipitate will be formed. So ketose sugar like fructose is present. So first concept is aldose reacts very slowly, okay, and give very faint pink color, which will helps in differentiating glucose and fructose. Glucose, the aldose sugar, reacts very slowly and will give a very faint pink color. It will helps in differentiating both the sugars, that is from aldose and ketose. Now, what are the some brilliant viva questions that examiner can ask you? So, why ketones react faster? They dehydrate more easily under acidic condition to form a furfural derivative. That's why. So, why should heating be time limited? Overheating can cause aldose also to react. So, it will lead to false positive test. Right? So, you have to be fast. Okay. So can sucrose give this positive test? Yes, sucrose can give this test positive, but the condition is required, and that we will be seeing in a next subsequent part after hydrolysis. Sucrose, when it gets hydrolyzed, okay, it will yield two products, that is one is glucose plus one is fructose. So due to this fructose, it will show the positive test. Right? I hope this thing is absolutely clear. Okay, so see this Saliminoff's test. Some ketones like fructose will see a deep cherry red color. See this. Some disaccharides also after hydrolysis will give this cherry red precipitate positive. Now you can see this aldol but very faint pink color. But we consider this test particular is negative because it is not a faint pink color. And control mein there is no test, no change in color. Okay, that's why the test. And this is how you can do the test. See, brightly cherry red color you can see. Okay, this test is positive. I hope this thing is clear. Now, second last test we want to discuss is the inversion hydrolysis test. Right? Inversion hydrolysis test we want to discuss. Theory behind the inversion hydrolysis test is a sucrose, which is a non-reducing disaccharide composed of glucose plus fructose, which is linked by anomeric carbons. So on hydrolysis, this sucrose will lead to glucose plus fructose. So this acid hydrolysis will ultimately lead to our invert sugar. That is a mixture of glucose plus fructose. So both are reducing sugars. So the specific rotation here of sucrose is plus 66.5 degree. But the invert sugar after the mixture is minus 20 degree. It is called inversion of rotation. Okay, inversion of rotation. This is called inversion of rotation. So what you have to do? You have to boil the sucrose contained sucrose solution with concentrated solution. of hydrochloric acid for 1 to 2 minutes. Okay, then you have to cool it down and you have to divide it into two portions. On a work pulsar, you are performing Benedict's test, and this Benedict test, this glucose upon hydrolysis of sucrose is give a positive Benedict's test. B positive लिख रहा हूँ मैं. Benedict's test is positive. And upon hydrolysis, the yields also fructose. Fructose will leads to Selimanov's test positive. Right? Positive Selimanov's. Okay. So red color precipitate. So hydrolysis confirmed, sucrose is present. Right? You can see what is the inversion of sucrose. Change in optical rotation. So it is called inversion hydrolysis. From plus to negative to hydrolysis of glucose and fructose. Right? So why sucrose become reducing sugar after hydrolysis? Because glucose and fructose both have free carbonyl group. Okay, that's why. How inversion can be detected? Polarimeter that can detect a specific change in a rotation. So you can see this. This is sucrose, right? I hope this thing is absolutely clear. And sucrose upon hydrolysis yields glucose which show Benedict's test positive and fructose will show Selimanov's test positive. Okay. Now last test in a discussion is a starch hydrolysis test. So starch is a polysaccharide compound which is made up of amylose as well as amylopectin. So when hydrolyzed, okay, when it hydrolyzed by acid or enzyme, it will yield maltose and glucose. It will yield maltose and glucose, and maltose and glucose both are reducing sugars. Okay, so what you have to do, you have to take 1 ml of starch solution first of all. You have to add 5 drops of concentrated HCl and you have to boil it for 1 minute. And then we have to neutralize it with sodium hydroxide. And then you have to add Benedict's reagent and you have to perform the Benedict's test on it. Okay. And then you have to see the observation. And once it got red precipitate, it will confirm the starch. Right? So on acid hydrolysis of starch, it will yield glucose plus mannose. Sorry, maltose. And this glucose will show Benedict's test positive. Simple test. Okay, so why does starch does not give Benedict's test directly? Because it lacks free group, free reducing group ही नहीं होता है, तो Benedict's test won't be there. What happens to iodine test as hydrolysis proceeds? So this, whenever the hydrolysis proceeds, blue color will weaken. Why? Because starch के अंदर bonds will be break down. So glucose and maltose will be liberated, and these two won't give the iodine test positive. ओके, बिकॉज आयोडीन को होल्ड करने के लिए हेलिकल स्ट्रक्चर चाहिए, वो हेलिकल स्ट्रक्चर ही नहीं है इन दोनों में. ओके, दैट्स व्हाई इट वोंट गिव दिस टेस्ट पॉजिटिव. So blue color will deepens down, weakens, and it will ultimately disappears when starch is completely broken down. So what type of bond broken during alpha? You can know alpha glycosidic bonds. So you can see this. Okay, you see this, right? This is a starch solution. And I have added a Benedict's test on it, Benedict's reagent. So it is showing a negative test, but whenever after hydrolysis I am doing the test, it will show the positivity. You can see. So glucose upon starch hydrolysis will show Benedict's test. This test is positive. Okay. So now let's discuss the overall summary table. Okay. Right. So Mollis test will be given by all carbohydrates. Positive result will indicate violet ring. And key reagent here is alpha naphthol and H2SO4. Iodine. So first is Mollis test. Iodine test is given by the polysaccharide, either blue, violet, and brown. Okay. Dextrin will show violet test. Glycogen will show brown test. I hope this thing is starch will show blue test. So key reagent is iodine, glucose iodine you can say. Benedict's is a reducing sugar will show the positive result will be from the green to brick red precipitate. Okay, key reagent you should know. Barford test will be given by monosaccharide and red precipitate at the bottom. Selimanov's test will be given by keto sugar and cherry red test. A cherry red color is positive. Inversion is given by sucrose. Benedict's and Selimanov both will positive after inversion. Okay. And starch hydrolysis, upon the hydrolysis of starch, they will yield glucose and maltose, and this will show red precipitate after hydrolysis. Okay, so what are the final conceptual viable questions that one should know? I have summarized all the questions. Okay, so you have to define reducing sugar. Okay, a sugar which is capable for reducing metal ions due to free carbonyl group is a reducing sugar. I am going quickly because you have discussed these particular things in your theory classes. Okay, non-reducing sugar which contains no free carbonyl group. Example is sucrose. Furfural derivative kata is dehydration product of sugar in acidic medium. What is the function of sodium carbonate in Benedict's reagent? It will provide a alkaline medium. Why iodine is reversible? Iodine complex is physically held. Upon hitting, it will release it. Okay, that's why the iodine test is reversible test. Right. So because in a helical structures, the iodine molecule will be held by the polysaccharides. If I hit that particular helical structure, they will disappear. So iodine won't be held, so color will be reversible. What is the difference between amylose and amylopectin? Amylose is linear, amylopectin is branched. Which test distinguish ketos from aldose? Seliminov test. Obviously, I have discussed this in very detail. So why Benedict's test is non-specific? Because it is detecting a non-sugar reducing agent also, like ascorbic acid and uric acid also shows this positive. I hope this thing is absolutely clear. So we conclude the practical here. And if you have any doubt, kindly ask in the comment section or you can approach me directly also. I hope this thing is very clear. Okay. We have covered the practical very well. Okay. Thank you for patience listening and best wishes to all. Okay. Thank you.